So for this workshop, we represented by Arno Sales, a student in the University of Engineering and Technology, Tech. He was working at Yale University, and his presentation is titled "Electroporetic Separation of Material Cells for a Fast Induction Solar Pumps."
Hello, congratulations everyone. We are doing the REPU Seminar 2017. So in this opportunity, I will explain our work part of the separation of material cells for a fast infection. So I belong to the branch of nano-REPU and currently work with the PhD candidate Sharisis in the lab at Yale University. So to begin with, nowadays there is a great deal of ways in which one person can be detect if the blood sample is infected or not. So that takes around 3 days. So because they are using mostly the cell culture techniques. So in order to speed up this process, there is a technique called that is using the LOC, the lab machine, that can speed up the process by from 3 days to 30 minutes. So let us explain how this will work. So this is a LOC, the lab machine. First we have the silicon substrate as the base. Then you have the electrodes as the electrical components, and then you have the microchannel that will guide the microfluid. So you can have the fluid in the shape that you want, and so this will give the micromechanical components. And then, of course, there will be a lid that can seal all these latitude. And this is just around 1 millimeter thick, so you will see that it's very, very tiny. So the advantages of this lab chip that you can do the lab tasks on one chip are that its portability, because it's very small, so you can carry it in very far away places. And also it's inexpensive, because what is properly designed, it can be massively produced. It's practical, because what is automated, you just need to put the sample, the blood sample there, and they can tell you if it's infected or not. And also is the consuming device, because it only requires like 1 millimeter of blood sample, and also takes less reagents. So that means that it is very practical and automated. So right now, this lab machine will be applied in the infection detection, and in this case, tuberculosis, because nowadays, sadly, we have the second highest rate of mortality in all the Latin America. So that needs to be changed, at least in the speed of the process. So our infection detection method relies on electromagnetics. So right now we have the same chip, the schematic, the silicon, the electrodes, more about complex work, the microchannels, and then the fluid. So in one inlet, there will be the infected blood. The infected blood sample, the sample that you want to detect if it is infected or not. And in the other inlet, there will be a buffer. I will explain that. And then there is a voltage source and analyzer that can connect these electrodes so they can measure signals and also analyze them. So at the beginning, you will put the red blood samples and also the material cells in one inlet, and the other one the buffer. So once they are going into the microchannel, the buffer will make these channels to be aligned in the left side, for example. And then once they get to the electrodes, thanks to that electric force, this will attract the material cells into the right side. And so thanks to the buffer, the material cells will continue into that direction. So thanks also to the microchannel, this all will be sorted and separated. And then there will be a part of the cell counter in which the cells will be counted. And that's how you can, in general terms, determine if the blood sample is infected or not. So right now we are using the data analysis, which will be done right now on the computer, and then you can integrate it in just one single integrated device, so you can do all the data analysis and all the analysis of the blood sample. And also, right now we will be testing with a bacteria similar to that tuberculosis, which will be Mycobacterium leishmaniasis, instead of using the same material instead. And right now I will focus on the election. So how this attraction of pure cells works. So we show the better preferences to attract the desired particles. So first we have, for example, 2 electrodes, negative and positive, with different shapes. And then right now the field is found. Then the fuel cell has, of course, problems on it. It doesn't matter exactly how it's charged or not. So when you set up the field on, you will put these non-uniform electric fields. And with that, all the protons and electrons will start like, having a polarity. And because of that, for example, the part that has the electrons, the force is said to be positive. That means that it will be attracted to the highest gradient. So in that case, it will be attracted to the right side, so to the positive electron. But for example, if you have a bigger cell, a human blood sample, it's bigger. Of course, it has also protons and electrons. But in this case, since this force is not enough to move it, because there is also a fluid, that won't be attracted, that in general terms. Also, so this cell will not be attracted, but the fuel cell will be. So this is a question of these force stresses. It is mainly dependent on the cell properties of the size, conductivity, and conductivity, and also mainly because of the fluid and for the geometry of the electrons. So this is a study work that was also collected the equine on the record cells. So you will see that these cells are flow with the field off. And then when this field will be like set to on, the fuel cell will gradually start being attracted to the electrons. So you will see that after 10 minutes, for example, these cells will be attracted, while the regular cells just falling around. So the advantage of this is that it's also, you can put it the other way around. So you just set the electrons up, and all the cells are just continue falling. So that means that you are not altering the composition of that. So that is good to remember about attract these cells. And that is that. Of course, you have to have all the chips completely sealed. Otherwise, the fluid can be like start leaking, and you will not have the proper data at all. And all the data you are like measuring will be lost. So this is it, and this is how the fluid should behave. But if there is not a bonded area, it will start leaking. You will not be able to perform the work as you were supposed to do. So this is how the unbonded area looks like before the fluid that has, for example, an air bubble there. It's called like a neutron ring because there's a fractional type. So then when the fluid enters, you will see how the fluid is just leak in all the area. So that needs to be implemented, of course. So in the literature, there are many methods to do that. Thermal bonding, which uses a higher temperature and pressure. The anodic bonding, which uses also voltage and temperature. The electric bonding, which uses also temperature, but it uses voltage, which uses like a metal intermediate layer. And the adhesive bonding, which can like bonds nearly off substrate. Because the other ones is mainly for silicon to silicon and glass to glass. But this is very useful, and that's like what we're going to use that. Because we can like bond SOL, which is a photoresist material we're going to use to give the microstructure. And also you can bond the glass substrate. Okay, so this is how the bonding sequence in general can also looks like. First we have the chip, we have the electrode with the microchannels, this is H2A. We add an intermediate layer, which is also this H2A, so it can be bonded with glass. And with that, you have the temperature and pressure, and now they're off. Then you set up the temperature to a desired temperature, let's say 90 degrees Celsius. And while you reach that temperature, you put the pressure. And then you put your pressure in the amount of time, it can be 1 hour or up to 8 hours, it depends on the materials. And then you start reducing temperature, and when it gets to the room temperature, you put out all the pressure. So this is how it's supposed to be bonded. This is like a general procedure. So so far we've been doing the chip level bonding, like 1 chip by time. And then the pressure has a range about from 5 newtons to 2,000 newtons, and that's in pascals, 600 to 300,000 pascals. And then the temperature is also around from 90 degrees Celsius to 210 degrees Celsius. That is also mainly dependent on the glass transition temperature on each material, and also in order to be in this bond. And then the time in which the pressure and the temperature was set on, it was around from 1 hour to 8 hours. So that means that in order to test the one chip, it takes an hour of time. So far we performed 32 tests, in which one test lasts around 2 days. So you will see how it goes. So this is how the outline of the layout of the bonding is like. So the first parameter is the pressure and also the temperature. So there is like a range of which we use. And also there is a glass and chip treatment, which can be the UV sun or oxygen plasma, or to put out the UV in a photo exposure. And also the detail layer, in this case, we use in SUA, sulfuric SUA, and also how the SUA is taking this sulfuric or hardly. And also if the chip substrate is silicon dioxide or only silicon. So there are many variables, there are many parameters, so we've been like playing with the parameters and also the variables, like of course with the theoretical background. And the results that we have so far that we can bond in this case was the oxygen plus exposure and also SU8 to SU8 bond. So I will show you how all these fluid did. So these two methods were the ones that work. So right now you will see that how the fluids are right now flowing into the microchannels. They are not linked at this time, but for you will see that they behave differently. In this case, the oxygen glass exposure, they behave like normal, but at least they have a little bit of flooding. That means that it is like interrupted to the water. And in this case, you will see that instantly, only in the last part. That is because there was a tiny particle there, and because that particle, it creates that neutron ring. And so there's an air bubble that all the water gets in. But that was only because there was a tiny particle. So if we are able to take all the particles away, if we're able to take it like very carefully, there will be no particles there, and then all the bonding will be able to be performed in some way. So the oxygen glass exposure, there's no need to add an analysis layer because there was no need to add an SU8 layer into the glass, but only like expose the glass with the oxygen plasma, because that creates filament groups that bonds with the SU8 in space. So, but the problem is that it is sensitive to the rough surface because the glass itself, it has a higher viscosity. And because of that, if there is like a 1 tiny particle, that area will not be bonded. But in the case of the SU8 to SU8 bonding, it has a low viscosity, it just flow it. So an idea like to take out that issue, we would be able to take out the SU8 that is above on the glass in the part of the microchannel. So with that, we can really stop flowing. And then it is not very sensitive to the rough surface because there is an SU8 to SU8. So now it can like tolerate something cardio, but because it is easier. But of course, it is ideal not to have any competition at all. But so far, from the theoretical tests, we found that these two methods work. So as a summary, bacterial cells can be rapidly sorted by dialysis processes with the of course the work that has been done with the inflate. And then this thought to be working with mitochondrial. And the accident lab machine depends on the parameters such as the temperature, pressure, chip substrate, amplification, intermediate layer. And these two methods, the oxygen exposure and the SU8 to SU8 bonding, they work. But of course, there's like a trade-off. And the oxygen exposure is very sensitive to a rough surface, but you don't need to add an aggressive layer, so you don't need to add like another application step. But in the case of the SU8 to SU8 bonding, you will have to first to add this SU8 into the glass, and that will be like more work. But at least it will be like easier to bond because it's not that sensitive to the surface because it has a lower quality. So with that, that is just the decision to make, and that's why in future direction we are gonna test how all these parameters can be optimized in order to try to use the wafer level bonding. If we're using one chip on one chip, we can use all the wafer can be bonded, and then cut them all the chips. So that can increase all the fabrication of all these chips. So we can then test the cell counter and all the parameters that these dielectric separation material cells so they can be optimized, they can be in the future used in order to detect time detection very fast. So I will thank in the first place Sharon Siski for being such a great advisor, and also all the REPU lab for welcoming me here, and all the lab members that have helped me a lot without exceptions. And also the Yale University for hosting me on all the paperwork. Also the REPU program that has given this such amazing opportunity. And of course, my mentor, Sophia Espinosa, for trusting me all the time. And also my home institution, FUTE, for giving this financial support, also for giving all the dedication that now I have. So thank you for your attention. I will be more happy to answer any questions.
Yes.
When you in your schematics of that you separate the one cell from the other, the first I think just first slide. Uh, do you have to have similar buffers, different buffers? What is the buffer? I mean, how do you play with the buffer and strength um, to separate cells? I mean, how do you use it? Because you have, your effective load and buffer. These are, I am assuming these are different buffer composition.
Yeah, sure. It's like recommend to have a buffer with large community. So with that, you're gonna like alternate all the electromagnetics that happens to electrons. So as someone has that connectivity, you will be able to keep pushing that's material cells once they are affected. So they can be so they can start following into the direction. And then of course once
it's the same, I guess my question is it the same buffer? Those two we have to dilute the sample in the same buffer so they.
Yeah, that's a good question. So that once the chip is properly fabricated, we will be able to test with all parameters in order to be in that moment right. So right now, of course, that sensitivity of the buffer matters because in the question that I prepared it also matters how is that contributed to media. So in that case, that will be the buffer. So as long as I have a lot of sensitivity, that will be able to to work as the way we are planning to do.
Technical question. What do you use to generate this animation?
I use Keynote, which is a PowerPoint of the Mac, but you have to get a lot of presentation.
Yeah, it's
The
reason I'm asking because in the assembly of experiments we do, we use automatically chamber that are connected and well, there's no electronics, but essentially.
Yeah, I know that it can be some problems to do that, but I just use this one because it's a very nice way to see it.
Yeah.
So people can understand.
So this is about the idea of this application. The question is it applicable to viruses? Can you trap viruses? The viruses behave the same and you trap them with the electric field. And the other question, at the end of the day, you will, with this analysis, you will know whether there's a bacteria presence, but you don't know what type of bacteria, right? Or are you going to be able to? Are you going to be able to? It's just a detection for infected and non-infected, right? If you don't know, then you don't have a clue of what the bacteria is. Will you be able to isolate a bacteria once it's detected?
Theoretically, any type of bacterial cells has a different shape, different properties than the other bacterial cells.
Also, you will the profile.
Yeah, so the cell properties, those parameters are set into the force coming to an equation of the direction force. So imagine if one bacteria cell has one diameter of 1 micron and the other one has 2. So you will be able to distinguish between these two. In that case, in kind of viruses, right now I would say that it might only work with bacterial cells, but I'm not really sure about the two effects. That would be a possibility.
So I'm curious about whether one of the final goals is to be able to bring this to a field or an area that has it's not accessible to labs, right? What are the applications of these? I mean, that's the goal. Is the idea to reduce this to sort different samples of how you're working with infected samples?
Okay, so the first of all, the thing is easy to answer because it's you can flush it via water and then when you just set the electric field to be off, all the parts will be away. So you can clean it as long as you have leakage. Okay, so that can be used. Okay, and also it matters the bonding strength of the of the lead input to the microstructure because it's like have a range of megapascal. So when you're like flowing, you're getting certain fluid, it comes, the chip has to resist all this pressure that is inside of it. So that's for one part. And the other part is I mean, ideally, when all these devices of the design and of the constructed, that will just be on the decisions of the same laboratory. I mean, if they're thinking, of course, it is ideal to have this chip integrated in just one device, so that device can be, you can like carry that device for quite a lot of places of people that do not have access, for example, to clinics or hospitals, can have these infections. But so that will be like a long-term goal. But of course, that will be everything.
How expensive do you do? How expensive do you do it now? How
Okay, usually in
can
the,
you get?
I'm not gonna, I can't give you like an exact number, but usually in all the fields of electronics, all the materials that are used in order to do that is very cheap because there's like these great fabrication advances in which makes you have spend less money to build this type of material, for example, silicon or electrodes. So one is probably design, you can put in the mass production, so you can reduce a lot of costs. But like an exact number right now, any other?
So something when you look at donation, are you doing the procedure in a clinic or is you say
In verification?
next year with donation you mentioned you have a particle.
Yes.
Possibly is what you're doing in a clinical, what kind of facilities you need to get treatment?
Right now, I'm trying to bond this to substrates in a lab. It is not very popular. Of course, one I can test and I can optimize the parameters, so we can know by certainty that this will work. Of course, that will be done. One big performance that work for the other one.
What is the current method of computation for working with the current method of computation?
Oh, so one of the most common ones is cell culture here. So you take sample of yourself and you like cell culture. So you can see like from that day, 3 days, with a 3-year flow, and then on the second if that sample was or not. So that's one of the most common.
So what's going to be the limit of detection for new systems? Can I allow to detect this area or is just a shorter time?
In a shorter time, I mean, instead of being like 3 days, it's potentially growing 30 minutes, and then around 30 minutes. And so with that, we kind of speed up the process much quicker. And also that will have a great advantage in all the detections and all the people that needs to work in a different context. So that that's a lot for the countries that have a lot of people. Any other questions? So if not, thank you for your attention. I hope you are enjoying this REPU seminar.
Thank you. So the booth is dominating from Prospect Street, so just, move for a couple of minutes and we'll let you through it. Thank you very much.
Maybe we have.
Una hora.
Parece ser muy limitado. O sea, como que más amplio.
Es increíble.
Eso es lo que todo esto y prácticamente realmente o sea, pero ayer avisaron de
Ayer analizado el decir, sacamos el marzo y de 3 meses ya dos personas ya estaban ya. Bueno, una es estudiante que se me ha graduado, otra bueno, mira, yo soy un poco de tienes que decir todos los días.
Oh, right, yeah. Yeah, not really. Like, I'll just go ahead, Greg. Yeah.
I work hard. Sí, She's yeah, she covers.
Okay, so, yeah, this is the one of the, it's like now they cost. The name is he didn't know what to do. So no one knew what to call it. They were like, very good. They said, okay, let's use it. And that's how, okay, our own people. I also was very good about like one of the I mean, it is like good, but not that. But I see a lot of people have I know for sure that
A mí me defensa. Debe de ser como persona. ¿Cuántas?
What's the name of the restaurant or hotel? Or.
Oh, wow.
Doesn't matter. I mean, right now I feel like I'm doing a lot of study that is yes, I know, like, how's, and it's also like these like brown. So this is really cool. But it's like it's such a fun person to go in Korea, you find such a here I have no idea. Like in Latin America. Yeah, it was. It's like every time you walk for us, which is like restaurant own sales like and you will be like your restaurant they want
We like
So in that usually 5,000. So for the house one per sale, I don't know if you also you are two marketers, concentrate on 2 or 3. Like the rest of us, I just want to go.
Yeah. So what we do is this, and you got
Sí, sí, en ese sentido que hablan. Claro, claro.
You like
Pero deberían usar, pero que deberían usar otra laptop, ¿verdad? Porque la mía, o sea, podría alterar un toque, el PowerPoint, o sea, las animaciones o algo. Le voy a pasar a Sofía. Bueno, al final de mi presentación.
Vamos a la de José. No quiero salir porque estoy cambiando. ¿Te escucha?
Hola, ¿cómo están?
A ti todavía está hablando, estamos enteros.
Ah, ya, no está trabajando.
De la comida. No, siempre es lo mismo.
¿A cuál? Por acá está mi laptop.
No solo está José.
Pero sí, que sí.
Pregúntale porque vamos a hacer.
Entonces, le paso no se vea.
Yo estoy preparado.
Pero primero que comer.
Que vamos a preguntar por esto. O sea, porque es normal, pero es normal que se puede alterar su favor. Porque sea
Te paso mi. Sí, exacto. Puedes pensar ustedes, aunque sí tengo pero
Apagan la cámara. Good that it was at the camera yourself.
Este es para. Esta es la cámara. La cámara.
¿Tienes internet ahí? ¿Dónde está la cámara que ven? Pero está en vivo.
¿Sigue transmitiendo?
No, sale como si estuviera silenciado el micrófono y la cámara. Pero no sé.
Ok.
Al Lupeche. Mándalo al Lupeche. ¿Cómo se llama? Un ratito, ¿verdad? Pasa. Ah, que te lo voy a mandar por correo. Mira que Ahorita por el trabajo. No eres
¿Te hice presión verbal?
No, no quería que me estaba solita.
Siento saltando porque se ve. Ojalá no. Y yo no. ¿Quién es el presentador? ¿Qué ahora con esto? Es con el VGA, sale normal. No, son VGA. Si no, probamos con él. Pero el HDMI, porque ya lo estoy enseñándolo ahora. ¿Cómo? Hostia, estamos empezando. No salí. Ya.
Simply, no se have to be 7 years of volunteer. I have to know it.
No voy a decir.
Good afternoon, everybody. Welcome back to the REPU Seminar. We're going to continue now with Gustavo Cano. He comes from the Peruvian University Agraria de la Marina, and he's been working here at Yale in the project called Enabling Efficient Carbon Nanotube Growth by Addressing the Drastic Decomposition. Let's give him a warm.
Okay, so good afternoon, everybody. Thanks for the introduction, Lydia. So yeah, I'm Gustavo Cano. I'm part of the REPU program. I work at the Plata Laboratory at Yale University, and today I'm going to be presenting this work that's entitled Enabling Efficient Carbon Nanotubes Growth by Addressing the Gas Feedstock Composition. So this presentation is going to be like this. I'm going to talk like briefly water carbon nanotubes, and then about the properties and applications, then about the synthesis of the nanotubes and their applications. And then how do we assess the problem in the laboratory? That's what you're going to be doing here, because likely I'm going to stay up to July, and then July, and the conclusions of this so far. So what are carbon nanotubes? To put it simple, they're just tiny structures of carbon that are cylindrical. As you can see here, this is a representation of a carbon nanotube. And you can consider a carbon nanotube also as a rolled up graphene layer. As you can see here, this is a graphene layer. It's made up of carbon atoms. If you roll it up, you have a cube, and this is what basically is a carbon nanotube. You can have a single wall nanotube, and you can also have multiple wall nanotubes. You have many concentric cylinders rolled up together. Carbon nanotubes of carbon, which means that there are another form in which you can find carbon in nature, such as graphite and diamonds. They are all made up of carbon atoms. The only difference is the way they bond, and this gives them special properties. So what are the special properties of carbon nanotubes? Well, this material is very important because it has a high tensile strength, a low density, high thermal conductivity, a large current capacity, and this is all given by the structure of the nanotubes. It's very important to consider that the way that the graphene sheet goes up determines many physical properties of the nanotube. Here is the chiral vector, which is what the direction of the folding of the nanotube, which is, as I mentioned, is very important because it determines many properties. And what you also have to have in mind is that the sp2 hybridization of the carbon atom that conform the nanotube is also very important because that bond gives you this, or basically gives you these three properties over here: the high tensile strength, the low density because it's a hollow structure, and the thermal conductivity because the heat is transfer to the preparation of those chemical bonds in the structure. So these unique properties allow us to make various applications for carbon nanotubes. Today, the most used ones are nanotubes as a part of composite materials or bulk materials, if you will. For example, for boat hulls or for any or for aircraft, they make these materials a lot less denser, but at the same time they are more strong. They are also used for water filters, and they have many applications in electrical fields because they can act as semiconductors or as metallic conductors as well. We also have various applications such as solar cells. As I mentioned, they form like a thin layer of composite that allows us a better transfer of both heat and electric conductivity. So I wanted to go through the synthesis mechanism of carbon nanotubes very briefly. So yeah, what this is the idea you have to have in mind. The synthesis, what you need is a feedstock gas, which is the input gas. You need a source of carbon in order to have the nanotube structure. So in this case, one of the most used compounds is ethylene, which is represented by this structure here, and this structure here is the same. So you have ethylene, and what happens is that you based on thermal decomposition, you heat up these gases, and then you get another also hydrocarbons. These hydrocarbons will react with this heated up zone, which in which the catalyst is sitting. The catalyst is in this case, it's an iron nanoparticle. So these compounds absorb into this iron particle, they dissolve, and then they precipitate to form this structure. But one of the problems today is that the composition of the feedstock gas is not really addressed. Not much study has been done in this field, and this is what mainly, one part of the study that's been done in my laboratory. So yeah, as I mentioned before, also the catalyst is very important to be considered into the synthesis process, because as you can see here, these are two different temperatures. When you have a temperature of 125 degrees Celsius here, and this is 825 degrees Celsius. And you can see that when you change temperature, the catalyst, the diameter of the catalyst or the size of the catalyst also changes. And this will determine the diameter of the nanotube. So this is another part of what you have to take in consideration when you're synthesizing nanotubes, which is another part. This is separate from the case of the composition, and we are addressing basically the case of gas in this case. So this is the method we're using is chemical vapor deposition, which I will be relating to CDD. That's how we call it from now on. So as I mentioned, all these applications are not currently available. They have not currently developed fully because there's a lot of knowledge in the actual mechanism of synthesis of the nanotubes. This limits the control of the CNT structure. This also limits the conversion and the carbon conversion and the energy efficiency. And this threatens these applications. So what we're trying to do is overcome these limitations to improve mechanistic understanding of the CNT formation or synthesis process. To do this, we're using a reactor, which is called a cold wall reactor. Unlike the general reactors that are used in the industry, they are called cold wall because they heat up the whole reactor in order to heat up the input gases and get a lot of species that will interact with this catalyst. But here, we're not heating all the reactor, we're just in this zone. And if you want, we keep the input zone as well, but not the whole reactor. So we don't get a lot of subproducts that we don't really need. For the experiments that we're doing, we're not using a thermal treatment source in this case. We have ethylene, which is the main feedstock gas, the main source of carbon. We have hydrogen that acts as a reducer for the catalyst, which is necessary for the synthesis. And we have this X, which will represent the different alkynes that we are testing, which are the test gases for our work. So what happens is that these gases go into the reactor, they interact with that heated catalyst, and they form this thin layer of nanotubes, which if you look with the microscope, this is a SEM image, a scanning electric microscope image. You can see these white lines, these are the nanotubes. And in real life, they look like this. That is CNT forest. It's really black, and if you take it out, it will look like a black powder. So here's work done by my laboratory today. Have determined that alkynes do have do enhance nanotube growth, and they have an implication in the nanotube synthesis. These three are alkynes over here: acetylene, propane or methyl acetylene, which is the same, and vinyl acetylene. These are not alkynes, these are other compounds that were also fixed into the reactor, but only with alkynes you can see an enhancement of the growth rate here. Yeah, this is the height of the nanotubes that we obtained and the time during the experiment. So the slope of this curve gives us the growth rate. And you can see it's better when you add alkynes to the mix of that. So as I mentioned before, we were using this gas composition, and the X here, the first one was acetylene, with partial pressure of 1%, 2%, 3, and 5%. We also used methyl with the same concentration. Vinyl acetylene, it was only used with 1% and 2%. And the other one option was when no alkyne was added to the initial mixture. And you can see here, this is the kind of results that we get from the reactor after plugging the data, with all the data into R. You can see here, this displacement is the height of the nanotube first. And for all of these that are the same, the time against the height. And from this graph, we can obtain the terminal height, the growth rate, and the catalyst lifetime. All these parameters are important to assess the synthesis of the nanotube. With this data, you can make these graphs over here. It's very important to have the growth rate because it gives us an idea of the kinetics of the reaction, which we're interested in. And if you can see here that alkyne concentration as it grows, for example, for acetylene, you can see that the growth rate is also increasing. The same happens with methyl acetylene. You see that it's also increasing, and the same happens with vinyl acetylene here. But when we have no alkynes added into that input mixture, you can see that the growth rate is really low compared to this other composition of the fixed gas. It is also important to mention here that we see a plateau over here. So if we keep adding, we are going to start more concentrations, but probably it's not going to make any much difference because there's like a limit already. So it's important for future experiments. There's not a clear relation regarding the terminal height of the nanotube and the alkyne concentration. And we will need to work in the show this in order to have a real trend here. So one proposed mechanism also that previous work being done in my lab is that these alkynes interact with the exogas, which is ethylene in this case, and they form a precursor of the nanotube. I mean, they attach to the nanoparticle, to iron, and they help to form the nanostructure. And that's why alkynes enhance that. You can see over here, there's another experiment done by my advisor, my coordinator. To prove that when these are the same text gases, acetylene, methylacetylene, and diacetylene, and you can see here that the main difference is the functional group. Here you have the methyl group, and here you have the vinyl group. And when you have the CNTs, the result of the CNTs, this is an excellent image. You can also see that these carbon nanotubes are well aligned. Well, when you use, for example, methylacetylene, it's the composition of the nanotube change. That's the morphology, and when you use vinyl, it changes as well. The methyl group is not very reactive, and we're thinking that it causes defects in another extract here. That's why the tubes fail. And here, the vinyl is as it has a double bond, it's easy for it to incorporate in the structure, and that's why we have like a less kind tube to say. Here it's another image that you can see clear. The methyl group makes the nanotubes more with more defects, if you would say. For future work, the main objective is to recycle a part of the fluent in order to assess the efficiency of the synthesis process. So this is what I'll be doing so far also, and what I'm going to be doing for the next month. I'm designing a recycling mission system, and then I'm going to assess the quality of the synthesis products produced, and also study the effects of the enhancers of the nanotube growth, as I mentioned before. So this is the schematic of the system. We have the input gas, the reaction director, as I mentioned before. We have some products, but some of them, we need some of them. For example, the aromatic compounds, we don't need them, so we're going to filter with a polyurethane foam filter, which is called a foam filter. Then we have a cryogenic to trap the VOCs, which are volatile organic compounds that we do not need, and they will interfere with our experiments. So after that filter, we can get the compounds that we want, and we think are going to enhance the growth, and then we're going to test it in the reactor. After that, of course, we need to analyze these gases, some of these gases, to see the composition of what's going into the reactor back again. And that's going to be done with our GC analysis, which is gas chromatography. To assess the CNT quality, the main technique used, and the easier one, and the most available one is the random spectroscopy. We assess the quality by comparing the composition of this nanotube with the sp2 carbon and sp3 hydroxide carbon. The sp2 carbon is the one we want to be in the structure because it's the one that forms the double bonds that I mentioned before, and this is going to be amorphous carbon. And the random spectra you get here, this figure corresponds to the sp2, for example, and this to the sp3, and we're going to compare the composition with this spectra. To observe the CNT forest, we're going to use scanning electron microscopy and also TM. SEM allows us to see the morphological aspects of the nanotubes, while TM allows us to see, for example, a closer look to the nanotubes, and we can actually see the number of walls they have. And then we also want to measure the conductivity and mechanical integrity allocated for the nanotubes. And these that we use for this are still to be determined. So that's what that's going to be done as well. So as conclusions, I just want to say that alkynes do play a role in the CNT growth. They enhance the growth and they possibly incorporate directly into the structure based on the SEM images obtained during the different tests of the alkynes. And the CNT growth mechanisms are yet to be fully established, and this is very necessary for assessing that efficiency of the process and to eventually get the application that I mentioned. And also the cold reactor, which is the one that we are using, is very appropriate for the study of this effect of the different compounds of in the phase of gas, because it allows us to control better the parameters inside the reactor too. Now I just wanted to thanks, of course, my PI of the laboratory, but the whole laboratory for giving me such a award receiving. Michael Yaneko, my PhD candidate, who is my advisor, and he's been very kind and helped me a lot through this process, and also Marco, my record mentor, who was always supportive. And this is all for the REPU program, which made all this possible and this opportunity. And of course, Leuven University for having such a great campus and a lot of opportunities for us. And that's all. Yes, thanks.
So the effect on the alkyne stars on the nature of the final carbon nanotube, with a single wall, a multi-wall carbon nanotube, is there any effect on alkyne stars?
Of the alkynes into it being
Yes,
single wall.
final product.
So the one way wrong that now are multiple.
All of them are multiple.
Yeah, the one we're obtaining are multiple right now. Because to obtain single wall, you have to also assess other parameters. Basically, the nanoparticles, which is the calories, you need to assess the size of them, which is by different temperatures or different calories. So yeah, right now we're only getting multiple.
Is the polyurethane foam filter, like, is that a known technique that it will separate out the VOCs and let the other gases pass through, or will you be doing that characterization?
Well, basically, it's a used technique to separate the PAHs actually.
Oh, okay.
So you trap them in that filter, and then you need to extract them. You do a simple extraction with some compounds, with the solution, and then you analyze them in a mass spec. Yeah.
You mentioned that the alkynes might be incorporated directly in nanotubes, so that could be one way that they're affecting the growth rate of the nanotubes.
Yeah.
Is there any other ways that this could also be helping, or?
Helping the growth?
Yeah.
Well, the work being done in my lab has said so far is that this over here is the proposed mechanism. Alkynes like incorporate directly to the nanotube structure. Thanks to this interaction with alkenes. So actually, we do need alkenes. Apparently, two in order to form this nanotube, and for these two, they are incorporated in the structure. So yeah, I don't know if that was another question for you. Did that help? Okay.
Well, continuing with the REPU seminar, now we have Luisa Zetra, who comes from Peruvian University, Cayetano Heredia, and she's going to be presenting today her work in Harvard University titled Elucidating the Mechanisms of Limb Regulation by PCBs in Power.
Thank you, Lydia. As Lydia said, I'm going to present my work of this winter, which is called Elucidating the Mechanism of Limb Regulation of Limb Development by PCB with Power. So, first, I want all of you to ask yourself, how can we be so incredibly complex? And I want you to remember that we all came from one cell. One cell that had to went through different cell divisions, cell movement, cell differentiation, that then lead to an embryo that had to develop different tissues and different organs, and that then lead to us with all of our complexity. So for me, this is why developmental biology is so exciting. My lab studies how our limbs develop, and so you must know that first, limb development can be divided into three phases. So the first one is limb birth initiation. In this step, when the embryo is 9.5 days old, in a certain region of the embryo, the mesenchymal stem cells will start to proliferate and migrate. So they will push the body wall and they will form this bag. This bag that in subsequent steps will start to grow and form the limb. This limb will then acquire its patterning in the three different axes. And in the last step, that is called tissue morphogenesis, this limb will become functional. It will become functional because now it will acquire the different kinds of cells, like for example, cells from the cartilage, cells from the bone, blood vessels, muscle, tendons. So I am highlighting this last step because my lab is working in trying to understand which are the mechanisms behind tissue morphogenesis. So I also want you to notice that at earlier stages, the limb is only formed by cartilage, and then the bone will start to be formed from the centers. So this whole winter, I worked learning how to perform correctly histology and molecular biology techniques. So I learned how to get from this a fresh mouse, and more exactly, I worked with the limbs to get a section of the limb that can be seen in the microscope with a better resolution because of the limb. So in this case, I'm showing you that humerus that is pointed with the arrow. And by having this, from this point, we can use this section for further assays as staining or molecular techniques with different approaches. For example, this is an immunofluorescence. So with these techniques, we can detect different markers of development and answer different questions. So I learned how to do H&E staining, in which in this picture you can see that eosine is in pink and is staining the cytoplasm and muscle fibers. And I also learned how to stain the whole embryo without having to section it. So in this case, I am staining the bone with alizarin red and the cartilage with ocean blue. So having the knowledge of these techniques, we can answer different questions of what is behind limb development. So to be more exact, what are the signaling pathways that are explaining how limbs are developed? So first, signaling pathway is the way by which cells communicate between them. And I'm going to talk about now the PCP limb pathway. So it is known that this PCP limb pathway participates in different developmental processes in which there is a tissue morphogenesis. So when this PCP limb pathway is active, it will produce an effect so that the cells will polarize or migrate, and this way they will shape the tissues. So this PCP wind pathway is present in invertebrates and vertebrates, but there is not much known about how this pathway is assembled in vertebrates. So my lab is working in this pathway, and it is known that in this case the ligand is Win5A, so it binds to the ROR2 receptor, and when it binds to the ROR2 receptor, it promotes the complex formation of ROR2 and BANGO2, which is another transmembrane protein. So this Win5A binding also induces the BANGO2 phosphorylation. And my lab demonstrated that this PCP Win pathway is involved in leaf development. So you can see here that when they inactivated ROR2 and BANGO2, two components of this pathway, there was not a proper leaf development. So PCP is planar cell polarity. So, however, as I said before, we are trying to decipher what are the components of this PCP Win pathway for leaf development. So, for example, I said that Win5A induces the phosphorylation of BANGO2, but it is unknown what is phosphorylating BANGO2. What kinase is phosphorylating BANGO2? So, to answer that question, a former member of my lab did an screening assay in which he knocked down different kinases, 480 kinases, so that he could know and identify which kinase was important for BANGO2 phosphorylation. So, for example, he was working with human embryonic kidney cells that had this BANGO2 in the cell membrane that is phosphorylated. So when he knocked down, if he accomplished knocking down the right kinase of this PCP pathway, he would expect that this phosphorylation would be gone. And he measured this phosphorylation by doing a Western blot analysis, because we know that when proteins are phosphorylated, there's a shift in the band. And by doing this screening assay, he found that STAT3 kinase was the one that could be having a role in the phosphorylation of BANGO2. So from this point, this is the proposed model of the lab, in which STAT3 is a component of this pathway. So again, when Win5A binds to the ROR2 receptor of the STAT3, that we then phosphorylate BANGO2. So we don't know if, again, this is certainly true. So we know that this is happening in vitro. So now my supervisor is trying to prove it in vivo, working with simple mutants and double mutants. So he has seen that when he works in simple mutants, when you mutate one component, you get a leaf defect. So now this winter I was working, they gave me a mouse line that had a knockdown in ROR2 and in STAT3. My results of that. What did I get? So these are my results. Again, this is the pathway, and what happened is they mutate ROR2 and STAT3. So if these two are part of the same pathway and are involved in leaf development, we would expect that if we mutate both of them, I will find something like this, a leaf defect, where you can see here pointed with the arrow that there is a leaf shortening, and there is not a proper formation of the bond. So in this case, this is a specific double knockout in the leaf mesentery. So I also did an immunofluorescence where I tried to detect a marker of leaf development. So in this case, I stained the hypertrophic chondrocytes with a specific antibody that stains collagen-10. So I stained them because it is known that these type of chondrocytes are important for bone development. So by doing this, they have a role they can tell us if there is a proper leaf development. So in this case, this is the wild type, and you can see that this is how it should look. We should have like a red zone, the hypertrophic zone. But when I worked on the double mutant, I found that this hypertrophic zone was gone. So this is why there is a disruption of the leaf development. So by doing these two assays, from these first results, I can conclude that this JAK-STAT3 is a component of this leaf signaling pathway, regulating the leaf development. So next, I would need to confirm this. I would need to analyze other double mutants of this same pathway. For example, mutating BANGO2 and STAT3. And also another thing that I was thinking about is that JAK-STAT3 is also active in different cells and regulating different processes. So I would like to know. So when I'm mutating this STAT3, I'm not only mutating the weak PCP pathway. I'm also mutating and knocking down the other pathway. So I would like to know if this other JAK-STAT3 pathway could be playing a role in leaf development. And that would be all. I'd like to thank my PI, Lindsay Jan, my supervisor, the Jan members, my mentors, the REPU program, my university, and Harvard University. Thank you.
Did you say whether it was an inducible knockout system, or is it a cell specific?
Yeah.
So every cell during development?
No, actually, I don't know. I mentioned it, but it's a conditional knockout. So they are knocking out these two genes only in the lymphocyte, in the early lymphocyte, by using the CRE system. The CRE system. So when you add CRE recombination, you induce a knockout. Yeah, because CRE would only be expressed in the lymphocyte.
Oh, okay. So it's a.
Because I'm using a specific promoter for lymphocyte. Yes.
You have the picture of the reservoir which was BANGO2 oscillation.
Yeah.
You said that the size difference is due to the phosphorylation, right?
Yeah.
But isn't it only the smear in the right band, the phosphorylation, and the proteins have different sizes?
Should you repeat your question?
And the right band shows us like smear in the upwards direction.
Yeah.
And is this the phosphorylation and proteins have different sizes?
So I think that in this case, for example, we're having these maybe because there are different states of phosphorylation. Maybe the same protein with phosphorylation in one side, two sides, three sides. So that's why we have a smear. So that would be explaining that.
Okay, but the proteins are kind of different sizes, right? So in lane 3, the protein was higher than the protein in lane 2.
Yes.
Is there maybe anything bound to the protein or something?
I don't really know. This is an example from, that I found. This is the case of a phosphorylated protein, P53. So I'm going to look about it. I don't know. So thank you.
Para conectarse.
Continue with the presentation. Now we have the volunteer, who is now an assistant professor in Massachusetts University. He did his undergrad in City College of New York. Afterwards, he his PhD in Cornell University as a postdoc study for them in MIT. He's also been awarded with a Service Scholar Award, and today he's going to present to us his work on the symbolic demands of energy.
Thank you, Lydia, for the introduction, and it's a pleasure to be back at the REPU, and very, very proud of you guys. You amazing presentation, very inspiring, very proud. And I'm going to tell you about the research that I'm doing in my lab. And please feel free to interrupt if you have any questions in the talk. But my lab, I started my lab at UMass Medical School in Worcester, Massachusetts, about 4 years ago. And it's basically it's a continuation of my studies that I did as a postdoc at MIT, where we try to understand how do cells respond to abnormal number of chromosomes. So as you may know, through evolution, all organisms have come to some type of evolutionary equilibrium where they have a constant number of chromosomes. This is the